Hi, everybody. Hello. Okay. So,
Tough.
can it hear it?
Barely.
Over there?
Barely.
Okay.
Should like
Okay, I'll come over there then. If you haven't seen me before, I'm Angeli. Hi. This is John. John is like veteran GOAT TA, like knows everything, knows everyone. So he's helping me out today. But we're gonna go super in depth, like this stuff. I know there's a bunch of names and scary things, and I promise it's not that scary. Okay, you just gotta know what the scary named things do, and you'll be fine. But just to start, how are you feeling after last exam? What went well? What didn't go well? Anything? Anyone? Don't know. We're actually just talking about this. I'm gonna say before finals. And an optimistic ideal world. I think so. Because the grading the finals is gonna take a while. Like a while.
She was here late last night trying to get them done.
Yeah, I'm sure soon. Very soon. But did it feel a little bit better at least going into the second one, knowing that you've taken the first one? Or just panic? Okay, just panic. Okay. What specifically, like, was it like the wording of the questions? Were you like doubting yourself because the first exam was like really stressful and now you're just like stressing about everything, or?
Everything.
Yeah, Gloria. Diagrams were good, the ones that she gave you on the test? Okay.
I think most of the questions, because at first I was like trying to answer it, and then I looked at the diagram, I was like, oh damn, all of the answers are right here.
Yeah, that was good. Okay, that's good. Diagrams helpful. Anything else? Anything good, bad?
I felt like I didn't have enough time to write everything.
Didn't have enough time, yeah.
Like such specific answers now. It's a half hour and I'm on the first page.
Yep, yeah.
No, but you know what Laura said? Laura said that if you need time, just go, like, just, if it takes more than 2 hours, like.
Yeah,
Go
so
for it.
we're never, and we were just talking
Yeah.
about this for the final, we're not gonna kick you out.
Yeah.
Laura especially is like very understanding if anyone needs extra time, or if you tell her that like, hey, I have so many exams on this one day, can I do like I think she's planning some makeup times right now. I'm not 100% sure what those are yet, but if you have like 3 trillion exams that day, like you can move it, like it's okay. She's very understanding about that, like test anxiety, anything. If you like need to take it at a separate time, or just like you need more time, like she's very accommodating to all of that. So, and yeah, you can also ask, like, I mean, John's everywhere. You can email me, like we all know Laura, obviously, so we can go talk to her if you need us to.
Legally through the university, if you have 3 finals scheduled for the same day, you can move the middle one.
Yeah. Like I remember, like I took her 285 class, and the final was like notoriously like took a very long time. And so it took so long that like we would do 2 hours in one room, and then we had to get up and move to another room because like you can only book the rooms like 2 hours at a time. And on my way, like I'm clearly like struggling when I'm like walking to the next room because I'm overthinking like everything. I was just like freaking out. I was like, this is not going well. And she like came up to me and she was like, hey, like, you okay? And I was like, yeah, I'm fine. So she's like, it's okay, it's gonna be done. And I'm like, yeah, you're right, it's gonna be done. Like she's very nice and she like cares about her students. So if you need anything, you can email me. My email is gonna be at the end of this presentation, and I'm gonna send these slides out anyways. So you can talk to any of us. But yeah, let's get into it. So let's talk cellular respiration. Okay. So what is the point of cellular respiration? So we have a bunch of energy stored in these bonds, and we need to use that energy. But our cells can only use the potential energy stored in like particular types of bonds, very specific bonds. So I don't know if you guys remember what you said in class, but it's like an energy currency. So, money is money, but I need to understand it in the way that it relates to me. So I need it in like American dollars versus other information needs to be in euros, So it's the same information, but I need it in a certain way. And that's what cellular respiration is about. And we'll get into it. So what is it? So cellular respiration, in a sense, is a very, very carefully regulated breakdown of glucose for us to get the energy from those glucose bonds to convert it to the energy stored in the bonds between the phosphates and the APP. If it sounds like a lot, it's okay. That's what we're here for. That's what we're going to go into in the slides. So quick way of saying it is you're breaking down glucose and you're translating that energy into a way that the cell can understand. Okay, so we have a bunch of energy stored in the glucose. We want to use it. How do we translate that into a way that the cell can understand and how to use it? So that's what we're gonna do.
Okay, so get started about the cellular respiration processes. It's important to talk very base level. So understanding the difference between catabolism and anabolism is very important. So catabolism is what we start with as a breakdown of major molecules. You don't have to really think about anything other than glucose at this point. But reactions for catabolism are reactions that break down very high energy yielding things such as glucose into smaller materials. And anabolic reactions or anabolism are reactions that need energy and they build different materials from other starting materials. And so you could think about like the formation of a different kind of like, I don't know, carbohydrate from smaller carbohydrates. But at this point, we just don't really need to think about more than it just building things up. Any questions so far? Yes.
As exergonic and endergonic.
We'll get to exergonic and endergonic in a second. This is more a little broad.
Yeah, it's just broader, like, are you building something up, or are you destructing it, basically, destroying it?
Okay, and this is just an example of catabolism. So here, glucose is gonna get decarboxylated, or you're gonna take out carbohydrates or carbon and carbon dioxides from glucose. And you're gonna, no, I said that wrong. You're gonna take out 6 different hydrogens, and you're gonna add it to different oxygen gases, and you're gonna form water. And in this process, your glucose is gonna get broken down into 6 different carbon dioxides, which is a gas. So basically, you're going from the bigger glucose into these smaller molecules and ATP, but we're going to cover the ATP forming process in a second. Okay, so now endergonic and exergonic. So endergonic reactions are reactions that are going to have a product energy that is going to be higher than their reactant energy. What that means basically is that for this reaction to happen, you need energy input. If you don't have an energy input, it can't happen strictly because the free energy, which is the energy level of the product and of the reactant, isn't compatible. So for a reaction to happen without energy input, you need the free energy of the products to be lower than that of the reactants. And for endergonic reactions, that is not the case, which is why you need the energy input. For exergonic reactions, however, it's the exact opposite, and it works in a way that you don't need energy input because the free energy of the products is going to be by default lower than that of the reactants. And so if you see at the graphs over there, they kind of look like little hills, and I'll explain that in a second. But basically, like we were talking about enzymes in the previous topics, right? For enzyme kinetics, what you can change in a reaction is the activation energy, or this little whatever you need over the energy amount in order for the reaction to occur. And enzymes can do that. So enzymes are catalysts for reactions. You cannot change the free energy. The free energy of the product and the reactant is not going to change. Is that good?
Yeah.
Okay.
I used to think of it too, like exergonic, I thought like exit, so like I'm releasing energy, and then endergonic, which is like the other one. And so what's really important to understand here is like what John just said, like in the enzyme unit, remember the reactants and the products, like where those guys are in their energy levels doesn't change. That's not what we're talking about here when we're talking about enzymes. Enzymes change the energy needed to make that reaction happen, usually by lessening it, right? So we want more reaction to happen. We're going to lessen that threshold that we need for that reaction to happen. And that's what enzymes do. But when we're talking about endergonic and exergonic, just think about where are we starting versus where are we ending. If we're starting lower than we're ending, that's endergonic because I'm building to get to the end. Exergonic, I'm starting higher, I'm ending lower. That's how I used to think about it. Does that make sense? Any questions?
Then I also added a little trick. And I was looking at this material more. Often I like to look at like the alphabet list, and if you picture it in like a vertical way, like A at the top and Z at the bottom, N and D are going to be higher up than X. And so I just like to think about like the N and it, mostly the D, not the N, of it being like higher, and then the X being lower, which means that the X, you're not going to need the energy input because the products are going to have lower energy. While for the D, you're going to have products that have higher energy. And so you're going to need the energy input. Okay.
Oh, yeah. So whether a reaction is exergonic versus endergonic, if you have a question that asks you, what is this reaction? I want you to look at where the reactants are in relation to where the products are, right? So I'm just gonna write it here because I'm closer to the mic. So my reactants are starting lower than my products. What kind of reaction is that? Is that endergonic? Are we building, or is that exergonic? Endergonic, right? Because we're ending higher than I started, right? And then it's the opposite for the exergonic reactions. That's reaction, that's product, starting higher, ending, I'm thinking exiting, right? Exiting energy, what's that gonna be? Exergonic, right? Does that make sense? Okay, so if you see a question that asks that, don't look at the middle, don't think about enzymes, just think about where are you starting versus where you're ending, okay?
Okay, so I kind of covered this a little bit early, but what we're used to talking about in the enzyme unit is the activation energy, which is the hump of the reaction line. For, as Anjali was just explaining on the board, for exergonic versus endergonic reactions, you're gonna just ignore that. As much as you want to look at it for that perspective for enzyme kinetics, don't. You're gonna look at where the starting point is and where the ending point is. And just a good rule of thumb for that is that you're not gonna have like a flat reaction because those don't usually happen. So one of them is typically gonna be higher than the other. You just have to identify which one's which. You start reactants on the left, products on the right, and you just see which one's higher. Don't focus on activation energy for this unit.
Okay, so knowing that enzymes change the activation energy needed for the actual reaction to happen, and it doesn't change the free energy of the reactants or products, can an enzyme make an endergonic or exergonic reaction or the opposite? So can the enzyme switch an endergonic reaction to become an exergonic reaction? What do we think? Any answers? I see some shaking heads. Anybody have any reasoning? Anything? Any guesses?
The enzyme really focus on changing the activation.
Yes.
Matter about products react.
Right. So you're not changing where it's starting and ending. With enzymes, you're just changing what's in the middle, like the threshold that you need for the reaction to actually happen. You're not changing the R's and the P's. You're not changing the reactants and the products. Okay. So when we think about enzymes, enzymes can't make the reactant start at a higher energy or make the reactant start at a lower energy. It just is changing what's in the middle. Okay. Okay, so let's go into the kind of bigger stuff in cellular respiration. So we're going to go through the ways to harvest energy from glucose in cellular respiration. And one is through redox reactions. And John actually has a little trick that is really, really helpful that he'll show you at the end of that. But let's get into it. So it's oxidation reduction reaction. So it's a transfer of energy in the form of electrons from one molecule to another when covalent bonds are broken and formed. So the molecule that is losing electrons is going to be the oxidized, losing potential energy. And the molecule that's gaining electrons is going to be reduced, gaining potential energy. He has a good memory way to not get confused with this. I used to just think of it as like reduced. I was like, oh, well, does that mean that's losing electrons? But I remember that it's like the opposite. But he has a way better way of remembering it, which he'll go over. And so for everything that gains an electron, that electron has to come from somewhere, right? Or losing an electron, that has to come from somewhere. So you're going to hear the term coupled reactions a lot. And I'm sure I think that's in the problem set as well. So what do we mean when we say coupled reaction? It means that for everything, like every molecule that gains an electron, that must mean that there's another molecule that just lost an electron because we had to get that electron from somewhere, or the opposite, right? If something loses an electron, that means somewhere else a molecule's gained an electron. Okay, so that's what it means by coupled reactions. They kind of go hand in hand with each other. Does that make sense? So how does this happen during cellular respiration? So we have a transfer of electrons from the glucose bonds to a molecule called NAD+. And she says this in class sometimes, but I'm just going to go over there, honestly, to draw it. She says, think of NAD+ like an empty bucket. Okay, so NAD+ is the empty bucket that's going to go along and pick up electrons. And when it does that, it turns into NADH. And NADH, think of it as like the full bucket. So we just filled up the NAD plus bucket with electrons, we've become NADH, okay? That's the difference between the two. So NAD plus is therefore at a lower energy state because it's the empty bucket. We don't have electrons in it yet. It goes and scoops up those electrons. We're at NADH, which is the full bucket. So that's what I mean by lower energy state versus a higher energy state, okay? So can somebody tell me what is being oxidized or losing electrons here, and what is being reduced or gaining electrons based on what I just said? Let's start with oxidized. So which one of the two, NAD plus or NADH, which one is losing electrons? Yep, more guesses, reasoning, anything? It's okay, guys, it's okay. So again, think of it, NAD plus like an empty bucket, right? Goes, scoops up electrons, becomes NADH, and now it's a full bucket. So when NADH picks up the electrons, right? So electron, it's now reduced. Reduced means gaining, right? Remember that, gaining electrons. It's now reduced to NADH. That's what I mean when I say full bucket. So in this case, NAD plus is being reduced to become NADH. And then the NADH goes to the mitochondria and it dumps those electrons because remember, NADH is the full bucket and into the ETC, which we'll talk about later. And now it's oxidized to NAD plus again. So, and John has a really good memory technique that he'll.
Yeah, so it can sound very confusing, it can sound very hard. I'm gonna teach you a trick and you're gonna be like, oh, this is silly. Okay, so how do I know what's what? You're looking at this molecule and it's like really hard to see. If you can see over there, there's like a double line over there that disappears right here. That's where the hydrogen is gonna go to. But there's a silly little image. What is that? Okay, it's an oil rig. So, oh crap, it's being covered. Okay, but basically the way you gotta think about it, oxidation is loss, reduction is gain. So every time something is being reduced, it's gaining something, and every time something is being oxidized, it is losing something. So if we're looking at our NADH molecule right here, if you're being oxidized back to NAD plus, it's losing that hydrogen, right? And if it's being reduced into NADH, it is gaining the extra hydrogen. So that hydrogen has to go somewhere, and it doesn't just like disappear from like anywhere. So it's basically, think about the two molecules like playing hot potato, and it's like, oh, I don't want this hydrogen now. I want this hydrogen now. I don't want this hydrogen now, and I want this hydrogen now. And it goes back and forth because these reactions can use happen. And so oil rig is a great like mental like pneumatic that you just think about like, oh, oxidation is loss, reduction is gain, when you're like, oh crap, which one's which? So yeah, so it's a good silly little way of thinking about it.
I'm also pretty sure that that's the same thing that he told, because I remember I asked him, was like, how the heck am I gonna remember this? But I'm pretty sure this is exactly what he told me.
Oil rig is really good. Yeah.
I'm just confused with when, like, I understand that, so NAD plus gains electrons from glucose? Like, how, what's
So-
occurring in like the cycle?
This is occurring, so there's two different processes where NADH acts or NAD plus acts, which is the electron transfer chain and the Krebs cycle, which we'll both cover. We're just using NADH and NAD plus as a example molecule. So you don't have to worry about that just yet.
Yeah, we'll go into
But oil rig is a great little acronym to think about. It's like when you're panicking, it's like, oh, oxidation is loss, reduction is gain. Okay. Oh. Or we don't want to work. Okay.
Okay, so let's talk about glycolysis. So I want you to think of glycolysis in just two phases. Just two phases, that's all it is, okay? The first one is the energy investment phase. So in the energy investment phase, we use 2 ATP. We're investing 2 ATP because we need energy to do the work of this process, okay? And it's also useful for keeping the glucose in the cell once it's phosphorylated. So what does that mean? Basically, when I phosphorylate the glucose, once it's in the cell, it basically can't normally diffuse out like it usually can without that phosphate group added onto it. We'll get to more of that later. I just want to like mention it super quick, but I have a whole slide of that. And also it's a negative feedback loop. Now, what you're going to see this on your problem set, you're going to see it everywhere. how is this a negative feedback loop? Can somebody remind me what a negative feedback loop is? Or even just what is a feedback loop in general? Does anyone remember?
Something happening like later in a process loops back around and controls something closer to the beginning.
Yeah, exactly. It's something downstream affecting something upstream, right? And the thing that separates it from being a positive feedback loop versus a negative is, let's say, something downstream causes something to happen upstream that causes less of that pathway to happen. Is that going to be a positive or negative feedback loop? Negative. If that downstream component does something to the upstream component and now more of that reaction is happening, is that going to be positive or negative? Positive, right? So think if it's going back, if it's more is happening, it's positive. I feel like that's pretty easy to remember. And then if it goes back and now there's less of that happening, it's negative. So that's what the feedback loops are, just as a kind of refresher. And so what is the negative feedback loop here? And there's a question about this on your problem set, which is the whole goal of glycolysis is to make ATP, right? All of this, we just want to make energy. So if we already have a lot of energy in the cell, do we need to make more from glucose? No, right? Because everything always comes back to in the cell, we want to conserve our energy and we want to use our resources efficiently, right? So if I have no need to make more energy in the cell, I'm not going to go through this whole painful glycolysis process. So the negative feedback loop is when there's already a bunch of ATP in the cell. It goes into it in your topic notes a little bit, but you remember that whole list of like kinase reactions with like the super long names that seems kind of confusing? Basically, all that's doing is it's pushing glycolysis forward. It's just like the cell signaling where the kinase affects another kinase, affects another kinase. So when we have a lot of ATP, we have a built-in mechanism on one of the kinases. It's like the phosphofructose kinase, that long one. That when there's a lot of ATP, it binds to the inhibitory site, meaning that kinase can no longer do its job. And if that kinase can't affect the next one, which can't affect the next one, is glycolysis going to happen? No. So it's a really cool mechanism because it only works when there's a bunch of ATP. It requires a lot of ATP to be able to activate that inhibitor site, to bind to that inhibitor site. So if we already have a bunch of ATP in the cell, we don't need more. We don't need more glycolysis. It's going to bind to that inhibitory site. And then that stops the whole thing because it can't affect the next thing, which can't affect the next thing, which means glycolysis isn't happening. I know it's a lot. We're going to go into depth on that. But does that make sense of like, okay, bottom thing is kind of affecting something, you know, because the end result is ATP. It's going back and it's affecting that kinase, which affects a bunch of other things. Kind of? Yes? Okay.
Which, like, where in the process of the glycolysis is that one thing, kinase thing that can stop it?
Yeah, I think it is like right before the fructose like 1,6-biphosphate. I'm pretty sure.
Here? Like, are you talking about this diagram?
Yes. Yeah, so I'm pretty sure it's, yeah, you see this right here? Actually, it's on the topic notes, but it's, if you look at that photo of the topic notes, it's the arrow that's going straight to the fructose 1,6-biphosphate. That arrow is marked phosphofructokinase, that long term. So that's what I'm talking about.
Oh, yeah, it's called PMK1.
Yeah,
That's the one that gets inhibited.
cool stuff. Okay, again, we're gonna talk about more of that in detail later, but I'm just trying to give you a general idea. Now we're in the energy payout phase. And in the energy payout phase, glucose is now split into two molecules, okay? And it produces 4 ATP. So quick math, we're putting in 2 ATP and we're getting out 4. How much net ATP is left? 2. 4 minus 2. So putting in 2, we're getting out 4. That makes us a net 2 ATP for each glucose molecule. Okay. And the net products are down here, the 2 pyruvate. That's in like that big diagram right there, 2 ATP, 2 ATP. We'll get into that. So think about currency conversion, right? Glucose is what we ingest. It's unusable as an energy source. ATP, we don't directly eat ATP, but we can use it as an energy source. That's the language that we're talking about here. We need to get it from glucose into something that our cells can understand, and that's ATP. Anything else? Okay. So You're gonna see the term substrate level phosphorylation. I think that's one of the things in your problem set as well, is what is substrate level phosphorylation? And it basically just means it's the ATP production where the phosphates are added to ADP to produce ATP. Do you guys remember the difference between ATP and ADP by any chance? Yeah, kind of. So ADP just basically, D stands for di, so it has 2 phosphate groups. ATP is T stands for tri, so it has 3 phosphate groups. It's all basically just saying we're making ATP by adding another phosphate group onto ADP. Make sense? So now we're getting 2, 3 instead of 2. That's all it is. So let's talk about Krebs cycle. So how do we prep for a Krebs cycle? We don't just go straight into it. We need certain materials. So how do we do it? So we've come out of the last step of glycolysis with pyruvate from the glucose. So we have so much energy still in those. We're still not done with this process. We still don't have the same language that we need yet. So how do we harness that chemical energy that we still have left? Yes. Yeah. Oh, yeah, sorry. John, you want to answer that?
What was the question?
Oh, so it's the substrate level phosphorylation.
Oh,
Like what specifically?
so it's basically when you get the ADP molecule and you add a third phosphate to it.
Oh, okay.
So it's different than, like, there's other kinds of ATP reduction that you don't think about. It's when you get like, so, excuse me, let me try.
Is this around like the enzyme and like they're just substrates? Is that why they call it substrate level?
Yeah,
You
so,
can also draw it there too, because I took the phosphorus.
okay, so so ATP has like base that we don't need to worry about, and then 3 phosphate, right? And it's gonna be here. When you cleave off this third phosphate, is like, this is where the most energy is stored for like your molecule. So when you cleave it off, a kinase will use the energy that's released from breaking that bond to do whatever it's doing, right? So you're left off. So you start with ATP, right? And then you end up with ADP plus of inorganic phosphate. All that means is that it got cleaved off. What this process does is it gets one of these ADP molecules that has been used up as ATP, and it gets a different inorganic phosphate and it adds them up together. And so you go from ADP plus the inorganic phosphate into ATP again.
Yeah, and you're gonna see PI too on your topic notes, and if you're wondering what PI means, that's exactly what it means.
So is that the substrate level phosphorylation?
So it's being phosphorylated, which means it's getting a phosphate added to it. So kinase phosphorylates whatever its like function is, that's happening to ADP.
So it's going from ATP to ADP plus inorganic.
That's just an example.
Oh,
That's just an example. So when ATP is used,
Mhm.
right, it goes into ADP plus inorganic phosphate. That's just an example. That substrate level phosphorylation, you're getting an ADP molecule that has been used up before.
Mhm.
You're getting an inorganic phosphate and it's being, actually, that's why. Do not use that. So you're getting ADP. A kinase is getting an ATP from somewhere else. It doesn't, you don't need to worry about where. And it's adding it to the ADP, making it into ATP again.
Okay.
Or we're just adding on a phosphate to ADP to make it ATP. That's all it is.
And so like, is that what you like call glycolysis? Like, is that like an example of substrate level phosphorylation?
Glycolysis is the overall process. Substrate level phosphorylation is what happens when, so can you go back? So you see right there that you start off with ADP, right at the bottom, right there, after the dotted line, you start with ADP, there's an arrow that goes in.
Yeah.
So that process is where you're making new ATP. So you're getting an ADP molecule, and during this process, you're making a new ATP.
So can we call that substrate level? phosphorylation?
That
Yeah.
ADP into ADP, yes.
Yeah, so like that too, where it's like pointing the arrow.
Yeah, and so that process for each glucose that you use is gonna happen twice. So if you use 6 glucoses, you're gonna come out with 12 ATP.
Oh, because, okay.
So it's happening twice.
Okay.
So you're breaking down, so a good way to look at this, and I didn't want to get into like this much biochemistry. Glucose, right there, you see those 6 dots? There's 6 carbons. You see pyruvate at the bottom? There's 3 carbons. You're splitting each glucose into 2 pyruvates at the end. So like when you get into more advanced like biochemistry classes, if you want, you're gonna see glucose be called like 6 carbons. It's just like 6C instead of glucose. And then pyruvate is just 3 carbons. All that means is that you're breaking 1 glucose into 2 pyruvates. So 6 divided by 2.
Yep, this whole thing is literally like all of glycolysis. Like this is what we're ending up with. The main thing is what he just said, each glucose is split into,
Pyruvates.
Yeah.
What was your question?
Where is this ADP coming from?
Used up ATP or just like other concentrations of ADP in the cell.
yeah, it's just there.
Yeah, this is happening in the cytosol, but the other processes that we're gonna cover happen in the mitochondria.
Any more questions before we move on? That was a good question. Okay. So let's get back to our Krebs cycle. So we have pyruvate now, right? We split the glucose into 2 pyruvate each. We have like 4 at this point, right? So we still have so much energy that we can harness and that we can use. So how do we do that? Pyruvate gets transported into the mitochondrial matrix. What does the mitochondria do? It's the powerhouse of the cell. Powerhouse of the cell. That's what this all means, okay? Pyruvate gets transported to the mitochondrial matrix. Matrix is just the space in the middle of the mitochondria. That's all you need to know. It's just like space. And pyruvate now becomes acetyl. How do you say that? Acetyl-CoA. And CO2 and 2-NDH are produced per glucose molecule. And this is an aerobic process, which do you want to explain what that means by aerobic process?
I wrote it down because it's important, but we're gonna explain why it's important in a second.
Okay.
Just keep it in the back.
So there are 8 reactions in the Krebs cycle. You don't need to know them. Okay, we're not going to go over 8 reactions. What you do need to know is what's made from the Krebs cycle. And here's what's made. 3 NADH molecules, which somebody remind me, is NADH the empty bucket or the full bucket? Full bucket. 1 FADH2, which is another bucket that carries electrons. Okay. 1 ATP produced through the substrate level phosphorylation, which just means we just added on a phosphate to an ADP and now we have an ATP. That's what that means. And 2 CO2 is produced. So each glucose molecule drives 2 turns of that cycle. Okay. So here's the purpose of Krebs cycle. We need to extract energy to use to make ATP in the electron transport chain, which we're going to talk about in a second, / the oxidative phosphorylation, that process. And more cellular respiration byproduct is created, blah, blah, blah. And I think in past years, she asked a question on this on the exam, which was like which relates to CO2, right? This is like your daily life. So when you're breathing out very heavy, like give me an example where you're like breathing heavy, like in what scenario? Yeah, exercise, right? You're working hard, your body's working hard, you're producing more CO2. So you can see here, because one of the outcomes of this cycle is like, okay, you're creating CO2. That's because your body's doing work. So she might ask you something on the exam that's like, oh man, he's breathing like super heavy. Like, why, like, what does CO2 have to do with this? The more work your body puts in, the more CO2 you're going to create, the more CO2 you're going to breathe in, breathe out, right? And I usually think of that as like, okay, when am I breathing like really heavy? It's like when I exercise or like when I'm doing like really intense work or, you know, like panic attacks, right? Like people are breathing in a lot of CO2 because their brain is going through so much work. So that's what I meant by adding the more byproduct of CO2 is created, because CO2 that's something that can be related very easily to our daily lives. So let's talk about electron transport chain. I think this is what has been scaring everybody. Don't be scared. We're going to break it down. It's going to be fine. So Krebs cycle occurs in where the mitochondrial what's the word? Matrix, just the space in the mitochondria, okay? Electron transport proteins, chain proteins, are in the mitochondrial inner membrane, okay? So Krebs cycle is happening in the big space. Electron transport chain proteins are on the inner membrane, okay? So those are 2 different places. So NADH and FADH2, which are what? The empty buckets or the full buckets? Full buckets. They're dumping their buckets, their energy into the ETC, okay? And in a series of oxidation reduction reactions, that energy is passed from protein complex to protein complex within the ETC. So what does that mean? The oxidation reduction, it means that for everything that's gaining an electron, something's losing an electron, right? So when I dump the NADH, when I empty that full bucket, I'm left with an empty bucket now. So was that oxidized or reduced? Oxidized, because I just lost a bunch of electrons, right? So that's what that means, series of oxidation reduction. When I'm dumping the buckets and the buckets are dumping to the next thing, they're each trading off on each other, right? Because for everything emptying, something's gaining. Anything you want to add
Oh,
to
no, I
that
was
as well?
looking at the chart.
Oh, you just, okay. And so energy is extracted from those electrons, the ones that are being dumped, right, to drive HP production, but not directly, and we'll explain that. So potential energy, let's think of it like a staircase, like this diagram here, right? Erase this. Think of it like a staircase. So with each step, I'm dumping electrons, right? Like, so like NADH or what is it, FADH2, right? Dumping electrons, something's gaining, something's losing. And for each step, energy is being lost, okay? But that energy isn't just being lost to do nothing. That energy is being used to do work. And we're going to talk about what that work is in a second. But at the bottom of the electron transport staircase, the electrons still need to be put into something. So we've traveled this whole way, we're dumping, we're accepting, we're dumping, we're accepting, we're dumping. So what is the final step of that? Where are all the electrons going? So we use oxygen, and this is really important. So when we talk about the phrase, the final electron acceptor in the ETC, if this is ever a question, it's oxygen. All the electrons going down the staircase, they're all ending up so we can dump it into oxygen, okay? And this is also why you need oxygen to live, because you need something to hold all of the energy at the end of this. And when you add the oxygen, the H plus ions, the H plus ions is like what we've been talking about, like the electrons, right? We get water. So that's why also water is a byproduct of all of this. So if you have a question like that, what is the final electron acceptor in the ETC? What are you going to say? Close. What's water made of?
Sorry.
No, it's okay, it's okay, it's okay. I like that we're thinking. Oxygen. Okay, so going down the staircase, at the very end, something needs to catch all of them. Oxygen is that thing.
Why do we get to live though? Like, why is that so important? Like, why without oxygen that they don't have?
You don't have a source of energy that you could use like ATP. So your cell needs constant amounts of ATP to like performance function. If you think about like your muscle cells, for example, not like just the muscles that you move, but your heart, right? What's causing the heart to beat? Exactly, you need the energy for the heart to beat, right? So if you don't have that energy, what happens? Yeah, you have like a heart attack and you die, right? You need the oxygen because it's the final electron receptor. I just wanted to make a little correction. The H plus aren't the electrons. The electrons are what's being held in the bond that you're breaking. And so that's being transported down as well. But the H plus that you get released and that gets put into water, you need the bond, like you need those electrons for that bond to be formed. It's not that the H plus are the electrons.
Sorry, that's why he's here. That's why he's here to catch me.
So at the end, it's forming water, right?
Yes.
Yeah.
So if you look at half an O2, a weird terminology, half an O2 plus two H pluses plus two electrons give you one water molecule.
Okay.
What that ultimately looks like.
You want this one? Or does that work? Does
No,
that
it's
work?
fine. So like a water molecule.
Yeah,
Damn it.
this one, sorry.
Damn it. Okay, so a water molecule looks like this, right? So it's like OHH electrons, right? If you're starting with like half an O2, which is a weird terminology, it's basically you're just starting with an oxygen that has electrons, right? But you need those two H pluses over there, plus the electrons in order to make those bonds happen for you to go from this, plus this, plus this to that. So you need those electrons in order to make those bonds, because they need to be held together, right? They're not just like free flowing and being attracted to each other for no reason. They need to be held together. So that's why you have the electrons. So it's like plus 2E minus, which is two electrons, and that gets built into a one. And this is happening over and over and over. Yeah. So the bonds are formed by electrons. So the oxygen carries its own electrons, but you also need more electrons in order for the hydrogen system.
We kind of okay for now?
So where's the oxygen like forming in there?
It's just around.
It's just there.
It's just
It's just there to catch all the
It's just
all
there.
stuff.
And it's being transported by blood. Like every time you breathe,
Mm-hmm.
it's being like flushed out of your heart. Because the blood is in the heart. It starts in the heart and then it gets sent out everywhere.
Okay.
Okay, so at each step, energy is lost by the electrons that will be used to do work, right? You mentioned that before. Every step, you're kind of losing a little bit of energy. So what is the work that it's going towards? Let's talk about that. So here's what the work the energy is doing. What it's doing is it's pumping the H ions out into the intermembrane space. So basically out of the mitochondria against their gradients. And this creates a proton gradient, which means there's more H plus in the intermembrane space than in mitochondrial matrix. I have a photo that I will show you. And this is ATP synthase. So all of that work, all of that lost energy is going towards driving those H plus ions out of the mitochondria, just outside. Okay, that's what it's doing. Does anybody remember which way does diffusion go? Does it go from a high concentration to a low concentration or a low to high?
I don't know.
High to low, right? So we're pumping a bunch of them, we're getting it out there, comes out the matrix side. Now there's more in the intermembrane space. That's just what that fancy term means, just outside the matrix. And there's way more H plus ions there. So diffusion, we know, goes from high to low. So the protons are unable at this point to diffuse through the inner membrane because they have to pass through, and this is what we call the ATP synthase first. Okay, so there's a bunch out there, they want to come back inside because that's where diffusion goes, right? So what that proton gradient does is it drives the movement of a small rotor in this entire big fun protein. And this allows ATP synthase to catalyze the formation of ATP from ADP plus pi. Remember how we talked about earlier is like the goal of this whole thing is we just want to make more energy, right? Substrate level phosphorylation is we need to add phosphate group on to make ATP. So this fancy phrase, F1 HPase unit purpose to produce ATP from ADP pi, that's this guy, this like fun thing right here. And how that starts moving, like a rotor, okay, this is a machine. How that starts moving is through all of the movement of this H plus ions going through this rotor, which we've just made using that extra energy that we had when we lost at each step. Okay, so that's how it's moving, is this proton gradient, which we've made ourselves. We did this. This doesn't naturally happen. We push it out ourselves. It has to go through here now. That's what's making it work. And this machine, I just think of it as like this machine makes ATP. Okay, this is, it's showing you right here. So it makes ADP plus the, what does it stand for again? I always forget. Inorganic phosphate making ATP.
Is it kind of like filtering through to make like ATP?
Yeah,
So
it's like a machine.
like we were just,
Yeah.
she was just talking about, the rate of diffusion from a higher concentration to a lower concentration is what's driving that. So you have like a lot of ions up here, and there's the matrix, so there's a barrier, right? And so like, if you think about in a hot day and you have like an iced coffee, doesn't the water go from the inside of the ice to the outside of the cup?
Yeah.
You know, that's a completely different process. It's just a visual, okay? If we go back like to the slide right here, so the proton gradient, what we were talking about for the energy loss or work is that, do you see how there's a higher concentration of ions at the top and there's a lower at the bottom? For every step that's happening, you're having a proton or a hydrogen ion that's at the bottom in a low concentration being shot up against its will back to the higher concentration. So this is driving this work because you're using this movement, you're using energy in order to perform this movement for the proteins to perform their function, right? So that's driving the production of new like FADHs and NADHs, right? But the diffusion against the gradient is what's causing this to happen. For ATP synthase, it's the opposite. So for ATP synthase, you're getting this concentration that's really high. There's none at the bottom. This shooting up, which is natural, which is what's supposed to happen, is what's spinning the rotor. So It's like basically have a high concentration here, no concentration here. It's shooting through the protein and it's spinning the protein, which then allows it to perform its function.
Right, so the first step is like we're using the energy lost in each step of the ETC to move these H+ ions, this is the membrane, out of here. Okay, that's what we need energy to do that, okay? The second thing is, now that we have a bunch of H+ ions here, which we've just moved ourselves, this is the membrane again, and we only have a few here at this point because we just moved everything outside. They want to go from high to low because of natural diffusion, right? High to low. But now we have our fun ATP synthase here, ready to catch all of that and make the ATP. So now it's going to go through here to get back in like it wants to do. And now we're smart because now we've just made ATP. Yes.
Is the creation of the gradient as well as like the ions naturally diffusing through ATP synthase, are those like simultaneously occurring?
So, do you know how so a membrane, that's a good drawing, but I would just do like a little one. So you know how a membrane has phospholipid tails and then the heads? So in a normal world, and it works for like cellular membranes as well, right? If you have a charged molecule, it cannot go through, right? And protons by default are charged, right? And so if it was like a steroid, for example, like a tiny steroid, it would be able to make its way through the membrane because it can just interact with the hydrophobic tails. A proton can't, right? And so you have the mitochondria here, and that's the inner membrane. I'm sorry, the drawing's awful. What's basically happening is that this membrane is like continuous, right? And so the important stuff that's happening is on the inside of this matrix right here, but the protons can't get through that matrix. And so they're all lining up right here, right here. There's a bunch of them right here. And the process of natural diffusion through the rotor of the ATP synthase happens this way. Right. And so, yes, this is happening simultaneously because these are made. The mitochondria isn't called the powerhouse of the cell, like as a joke. Like you need ATP. So like if you think about like brain function, muscle function, like literally everything else you do in your life, you need these molecules to be made in order for you to have cell function. And so the way they line up is strictly because of the hydrophobicity of the membrane. But it's necessary, and this organelle is like specific for it because you need it. If you need like constant neural activation because you're thinking, right? You're trying to study. Think about the levels of ATP you're using in your brain just to think, right? Just for those neuronal signals to happen, right? Or like muscle function, your muscles don't care. Like you're tired, sure, but you're still like telling them to move. Or like the muscles that don't move because you tell them to move, like your heart or like your liver, your stomach, right? They're still moving. They don't care. They need to do it and they'll just keep doing it. And that's why you have a lot of these.
Okay, yeah, that was great. So if oxygen, remind me what oxygen is called. It's the final what? Yeah, final electron acceptor, right? So let's say we don't have oxygen. Is the electron transport chain gonna work or not? What do we think? No, right? So what do we do in that case? That's when fermentation comes in, okay? We don't have oxygen, we need something else, we need energy, we still need energy. So what happens when there's not oxygen available in the ETC? Okay, so fermentation is basically the process of taking the electrons in the NADH, empty bucket or full bucket? Full bucket, right? NAD plus is the empty, NADH is the full. And dumping them into pyruvate, which you guys remember we made earlier in glycolysis, to produce lactic acid, or other stuff we'll talk about later, or a downstream metabolite of pyruvate to like ethanol or something. So basically, we just need NADH to dump electrons out so we can regenerate the NAD plus, which is the empty bucket. And then these electrons from the NADH are dumped into pyruvate to create lactic acid. So instead of NADH going to the ETC and dumping and dumping and dumping, we can't do that now because we don't have oxygen at the end. What NADH is going to do instead is it's gonna go to the pyruvate that we made, and we're gonna dump it into there instead. So what happens when we do that? Questions so far? Fermentation? Okay, we'll get more into it. So, you wanna talk about this one?
Yeah. So I think a cool little way of just like determining what makes what. So humans are producing lactic acid. And so like, you know, y'all go to the gym, y'all like pick up weights, and if you do it for too long, you start getting tired and your muscle doesn't want to do it anymore. That's because your lactic acid is forming wherever the muscle is. And so like if you're doing like your crazy reps for like your butt, like your bicep curls, right? Your bicep's gonna get tired. It gets tired because of this lactic acid formation. And it's interesting to think about too, like just like a casual, like something that happens in your everyday life. You might have heard that yeast produce ethanol through fermentation. This act of producing ethanol is also releasing like carbon dioxide gas. And it's why we use it for like dough fermentation, for example. So like when you're proofing your sourdough at home, everybody had their COVID like dough making
You
phase.
made it recently.
I had made one yesterday, actually.
Yeah.
You need the sourdough to proof, right? And what's happening is like the yeast are like getting all of this, releasing it as carbon dioxide and ethanol. Yeah, or ethanol is alcohol.
Oh, that's why it poofs.
And it's growing.
Oh.
And so like if you do like a 12-hour proofing session, which is what you're supposed to do for sourdough, and you like punch it, it like mushes. And that's because like you're releasing all this gas. So it's like really cool. It's like this like thing that happens in your daily life that you don't even think about. So like next time you think about bread, think about the yeast that's, you know, fermented.
Yeah,
And
I saw a sourdough, it looked really good. He didn't bring me in.
I can bring you. And it's also like, you know, for alcohol production and like what you'll learn in like Orgochem lab if you take it, like fermentation is how you get alcohol out of like potatoes or like, you know, not yeast, wheat or like, yeah, literally anything that like any alcoholic beverage is produced by fermenting a grain and it releasing alcohol. Literally anything you ferment, like pickles and like kimchi, if you eat kimchi and like things like that, like that's fermenting through like this process, which is really cool too.
So the only difference here with fermentation versus that whole ETC thing that we talked about, at the end of glycolysis, instead of going to the ETC chain, which we can't do because we don't have oxygen, we're just gonna go and dump into pyruvate instead. And so we get, so lactic acid, it's never as good as like the real thing, right? Like we're not making what we actually want to make, which is like the best energy that we can, but we're making something because, you know, we don't have
What's
oxygen.
really neat, and you don't need to know this, and it's just like a little off topic, your body can use this, and acetyl-CoA and pyruvate are like centralized molecules, and they're involved in a lot of different processes, because you can use the energy that you're producing by making NADH and FADH plus or FADH2 and convert that into ATP production. So when you do have access to oxygen again, then you can use that like those like electron heavy molecules and use it to make the molecule that you really want. So that's pretty neat too.
Okay.
Yeah.
Yeah. Okay, that was it. I hope this helped a little bit at least. It's a lot of information. I'm gonna send out the slides. But I'll stick around in case anybody has any questions. Yeah. This one?
Yes.
Okay, yeah. Yeah.
Yeah.
So is this glycolysis, like what's the difference between this and like I know glycolysis, so basically like if there's no oxygen, Krebs cycle and oxidative phosphorylation doesn't occur. So but glycolysis does not the same as fermentation.
Glycolysis is the process of getting the glucose into pyruvate. Oxidative phosphorylation is the process of getting these molecules and transforming them into ATP. Fermentation is the substitute version of oxphos that's used when you don't have oxygen.
So like when you don't have oxygen, you do glycolysis and after.
No, you to perform. Yes, you're right.
Then
No
after-
oxygen, yes.
With that pyruvate you made with glycolysis, you do that.
Yes. So you need pyruvate in order for fermentation to happen.
Yeah.
But it's less useful. Not useful, it's less,
Efficient?
yeah, efficient's a good word, than oxphos.
Yeah, like if you're working out and you know, you suddenly get a bunch of lactic acid in your muscles, it's not the fun. Like, it's not fun, like it's not giving you more energy, right? And notice with these two, like you go glucose to 2-pyruvate, that's in both types of fermentation here, right? Which tells us that that first step is the same. So we're still gonna go through glycolysis to get to the 2-pyruvate. What happens after that is what's different in fermentation. In fermentation, instead of going to the ETC and dumping the bucket, we're gonna dump that bucket into the pyruvates instead of just going to the ETC like normal. Does that make sense? The electron transport chain.
What about the Krebs cycle?
Krebs cycle still happens.
The Krebs cycle can only happen when you have oxygen.
Oh, wait, really?
Yeah.
Wait, I lied.
So
Oh,
it's
yeah, yeah, yeah.
anaerobic. So here's what happens. You have glycolysis. You have two options after glycolysis, and so you use one glucose, right? How many pyruvates are you gonna get? Two. So you have those 2 pyruvates. Do you have oxygen? No. If you have no oxygen, then you go to fermentation. So we can
Yeah,
make like a
that's my
simple little flow chart. So glycolysis. What? Glucose goes and becomes 2 pyruvates.
And, okay, cycle.
Yep. When you have oxygen, then you have ETC and oxphos.
And Krebs.
Well, ETC and
Krebs,
Krebs.
yeah.
And then if you have no oxygen, then you have fermentation.
How is ETC and Krebs the same thing?
It's just a different name. It's the same.
Is
Or
that the prep?
no, I thought about CAC. You're right, Krebs. But ETC is Krebs cycle is involved in ETC. So if you want to centralize it, you can. If you want to call, so there's 3 different names for this Krebs cycle. There's a Krebs cycle, the citric acid cycle, and the TCA cycle. They all mean the same thing if you read like a source online. It's all part of this. So CAC,
So is the Krebs cycle overarching or is the ETC?
ETC. Yeah, because ETC involves, well, it's the name for it, but like it involves the Krebs cycle production of the electron carriers and the formation of ATP. So like it involves, so ETC involves the citric acid cycle or Krebs cycle or whatever you want to call it, and oxphos. Oxphos. Good book.
Any other questions? Okay, did I see a hand earlier?
Yeah. Do we in fermentation, do you have to know exactly how much of all the exact molecules or
That you get out of it? I would say know that, like, number one, that for each glucose molecule, you'll make 2-pyruvate, right? And if you look here, you can see that for each 2-pyruvate, you make 2 lactate. So it's just like a 1-to-1 kind of thing. But it's really important to know that for each glucose molecule, you're gonna make 2-pyruvate. Anything else? Yeah, I'm sure she's gonna give you guys like more diagrams as well later on. Anything? Does redox reactions make sense, kind of? Yeah, empty bucket, full bucket.
What does that happen?
Redox reaction?
That's in good.
All throughout the ETC.
Okay.
So it's basically a game of hot potato. It's like, oh, I want this, I want this, I don't want this, I want this. And like you need to be able to carry that somehow. And so it's just like one molecule giving it to the other molecule, waiting for it to come out.
Yeah, it's just a fancy way of saying like for everything that gains, something is lost, and for something that's lost, something else is gained.
Yeah.
Essentially. And remember oil rig. Remember oil rig.
Is there any specific number of ATP that is?
I don't know for this class, for biochem class, you just say that overall it produces 32. I don't think you need to know.
Yeah, I don't think you do, but
She did mention like 20 to like 30-ish
Yeah, and then you also convert you like convert the other electron carriers into like ATP, but I don't think you.
Yeah, I think you're fine. You know that it makes ATP. That's what's important. Real quick, before you guys go, really important, something I mentioned earlier. Why is ATP investment needed in glycolysis in that first thing, where we talked about phosphorylation? Does anybody remember?
There's a hand.
Prevents glucose from being able to sell.
Yes, yeah, literally exactly. So the phosphorylation of glucose keeps it in the cell and destabilizes it, so that's easier to cleave. Okay, so when we get glucose into the cell, we phosphorylate it, can't wait. We want to keep the glucose in the cell. We need the glucose. We need to make stuff. Okay. All right, we'll stick around. Thanks for coming, guys.
Thank you.
Yeah.
Stop the recording. We can start rendering.
It's for holidays.
Of course.
No, seriously.